Hello, I'm Georg Wölflein and I'll be presenting HoechstGAN, virtual lymphocyte staining using generative adversarial networks. This is joint work with Inwa David Harrison, and Ognjen Arandjelovic at the University of St Andrews. Let's first define some key terms. Staining is the chemical process of visualizing specific types of cells in a tissue section using artificial coloration. On the other hand, virtual staining poses an image-to-image translation problem, where we task a deep learning model to transform a whole slide image from a source stain to a different target stain, so the resulting whole slide image appears as if it had been stained with our target stain instead. Whole slide images, like the one shown here, are digitally captured tissue sections from under a microscope. They are in the order of 100,000 by 100,000 pixels in size, which is why we split them into small patches. Here we see the same whole slide image, but with a different stain. We study a special case of virtual staining, where we want to translate from one input stain to two different output stains. Our work is concerned with clear cell renal cell carcinoma, which is the most common type of kidney cancer. Here we aim to translate Hoechst staining, which highlights all types of cells in the tissue, to cluster of differentiation 3 and 8, known as CD3 and CD8, which are T cells and cytotoxic T cells respectively. We propose a novel GAN architecture to solve this task, alongside a new evaluation metric to evaluate virtual staining tasks in general. We will also make our dataset available as a benchmark for future research. Let's begin by looking at the simpler task, where we just have one output stain. Here it's CD3. This is the classical image-to-image translation problem, which can be solved using a convolutional encoder and decoder with skip connections, which is known as the UNet architecture. Seeing the UNet as a generator and adding a discriminator to this model forms the well-known pix-to-pix GAN architecture, which is just a GAN conditioned on an image. Our model, HoechstGAN, extends the pix-to-pix architecture to handle two output stains instead of one. Let's look at how each output is generated in turn. The top half of the diagram is responsible for CD3 generation, and actually this is exactly just a pix-to-pix model. The interesting part is in the bottom half. Notice first of all that the encoder E1 is shared across both branches. This incentivizes the encoder to learn a latent representation that is mutually beneficial for both output stains. However, notice that the CD8 decoder, which is labeled D2 in the diagram, actually obtains a concatenated latent representation. This consists of not only the encoded input patch, but we also encode the generated CD3 patch and feed that into D2 via a separate encoder. Finally, we modify the typical GAN loss and objective to suit our scenario. Here are some qualitative results from 5 randomly selected patches in our dataset. You can see that the generated outputs, which are labeled as fake in the figure, are visually quite similar to the real ones. We devise a metric which we call the relative masked intensity ratio that allows us to assess virtual staining quality, which are the numbers shown to the right of the pictures. This metric also allows us to perform ablations of our model and compare it to two different baselines. The ablations are marked with suffixes in the table, but our final model is in the top row. The bottom row is a regression baseline where we employed a UNet without a discriminator, and the penultimate row are the results of training two separate pix-to-pix models, one for each output stain. Our described model outperforms both baselines. Thank you for listening. Code, data, and more information are available on our project page.